Hello, hi, welcome back to a lecture. Actually, it is not a lecture today, but we are going to discuss something or we are going to solve some previous year questions. In this lecture, we can solve 50 previous year questions which were from the chapter of genetics, inheritance, or particularly from the chapter inheritance of variation. Okay, so I hope before you see this lecture, you should go to my previous 3, 4, 5 lectures of genetics and complete it and come here. I have discussed most important things in my lecture, which is needed for the purpose of solving the questions. Actually, when I went through the questions, I have noticed that most of the questions in the genetics are direct questions and easy questions. Plus, if you have read the textbook, which means you have studied the concepts, it will be better for you. You can easily solve the question. Actually, for genetics, solving the question is necessary than any other chapter in biology because genetics leads have a conceptual basis. So, understanding the concepts and doing the questions is necessary. It is not that you have just done the questions, because some of the questions here are direct, but some of them are very conceptual. So, in order to understand it, you should do it, study it. Okay. So, we can go to the first question. I think the question is visible for you. Okay. Just we can complete 50 questions in a matter of time. Very easy. Okay. Now, please listen. Question number 1. The production of gametes by the parents, formation of a zygote. Okay. The F1, F2, which means filial 1, filial 2, and it can be understood easily from a diagram called. We know that it is Punnett square. Reginald C. Punnett. He was a British genetist who made or who actually from his experiments he have concluded or made a Punnett square from which we can easily find the production of a gametes by a particular parent and a other parent. Yes, look, actually Reginald C. Punnett in 1917, okay, in 1917 he made or he done certain experiments, and for that experiment as a mathematical tool he made a square which is a Punnett square. So we know that actually that there is nothing much more to be discussed. Punnett, we will be having 2 rows here one column, here one row. So in this position we will be writing the gametes produced by a parent 1. Here we will be getting parent 2. We will be writing the gametes produced by parent 2. So in each of the column we can draw the multiplication, or we can fusion we can write the fusion of the two gametes of P1 and P2, right? So from that fusion we can understand the genotype and also phenotype of an organism. So from a Punnett square, which was founded by Reginald C. Punnett in 1917, he made a Punnett square in order to find the genotype and phenotype of an organism. Phenotype we can predict. Okay. So option number C. Okay, option C. Next, the question number 2. The number of characteristics, contrasting character. Contrasting means 2 different types studied by Mendel for his experiment was number of contrasting character. We know that 7 contrasting character were taken by Mendel in his experiment. Okay, he have done his experiment in a pea plant Pisum sativum having a diploid number of 14. 2n is equal to 14. Okay, next, so the option is D, 7. What were the contrasting character taken by Mendel? While I was taking my lecture, I have told you to remember a plant where the plant was having axial flowering. The flower were violet in color, having a green pod, a green inflated pod with round yellow seeds. Okay, these were the characters. The plant was also tall, a tall plant having axial violet flower bearing a green inflated pole with yellow seeds. All the sentence I have mentioned is having 7 contrasting character with their dominant genotype. Okay, the dominant phenotype. I think you have understood it. So option D, 7 is the number. 7 different characters were taken by Mendel. How many true breeding plants? Now question number 3. How many true breeding plant variety did Mendel select as pair, which were similar except in one character with contrasting trait? Okay, yeah, I think you have understood this. Please read it once more. How many true breeding pea plants did Mendel select as pair, which were similar except in one character? We know that Mendel took 7 contrasting character. So while he was doing an experiment, he took one contrasting character, which means that the selected male and female plant, okay, the selected male and female plant were having all the same character except in one character, which is the contrasting characters. What were there? Example, he took 2 pea plants. Every characters was same except their height, which means one of the parent was tall, one of the parent was dwarf. So he took 7 contrasting character, which means during a cross he should take 2 parents. So 7 into 2, 14 parents were, or 14 plants were taken. They were having same, each pair was having same except in one character. Okay, contrasting trait. I think you have understood it. So the option will be 14. Okay, next. Question number 4. Identify the wrong statement with the reference to the gene I that codes ABO blood group. Okay, we know that I have discussed it earlier. ABO blood group means it is having A type, B type, AB type, and O type blood group. It is a example of codominance. We can say that codominance comes under multiple allelism, where a gene will be having more than one allele in a population. Okay, I think you got an idea, right? Because I have said it very clearly in my previous lectures dealing with genetics, first chapter. So please read the options. A person will only have 2 of the 3 allele, right? It is correct, because in a population a gene may have more than 2 allele, but when it comes to a individual of the population, he will be carrying only the 2 allele of the 3 combination of the 3 different allele. Okay, that is correct. We need to find the wrong statement. So the option number B is, when IA and IB are present together, they express same type of sugar. Option C. Please go to option D. Gene I, which means I has 3 alleles. That is correct. So we know, we can identify that option 1 and option 4 are correct. I don't know whether you know about the sugar idea of sugars in RBC. I will give you a small idea. In an ABO blood group system, there are 4 types of blood group: A blood group, B blood group, AB blood group, and O blood group. 4 type of blood group in a ABO blood group system. Later it is classified on the basis of Rh positive, Rh negative. I am not going through that. We are focusing on the genetics. So ABO blood group system have the presence of 4 type of bloods. What is the idea behind the presence of these classification? We know that in genetics we have studied ABO blood group system as an example of multiple allelism and codominance, which means that in the 3 allele of a gene coding ABO blood group, there are 3 alleles: A, B, and I. I have already discussed this earlier, but I will say it once more. Look, if IA and IA are present, it codes for A blood group. If IA and small I is the recessive form, it codes for A blood group. If IB and IB, it code for B blood group. Okay, B blood group. And if it is in the case of IA, IB and small I, it also codes for B blood group. If it is 2 small I, which is the only recessive form here, it will codes for O blood group. And if it is IA and IB, which means the two different forms of the allele, dominant forms of the allele are present together, they will form an entirely different blood group, which is AB. This is the case of codominance in multiple allelism. Multiple allelism means more than two allele present in a population of a same gene. So look here, in this condition, it is an example of codominance. Why it is an example of codominance is because if both different dominant allele are present together, they will be expressing together, forming an entirely different genotype. We know that in case of codominance and intermediate dominance, both the phenotypic and genotypic ratio are equal. 1:2:1 is the phenotype ratio, 1:2:1 is the genotypic ratio. We have discussed it very clearly in the lectures earlier. So IAIB. So this is an example of codominance. So how the dominant allele or the recessive allele leads to this change in the RBC, right? RBC is the red blood cells. So how the expression of this allele changes the RBC or changes the blood group in a particular organism. I think you are getting an idea. You are having any sort of doubt, you should ask me in the comment box. How the expression, how the dominant form or how the recessive form will make the change in a red blood cells so that it becomes a blood, B blood, ABO blood and O blood group. For that, you need to understand the structure of RBC. RBC or red blood cells contains, they are a cell, they have a lipid bilayer. Just understand it. I will show you structure, then you will get a more understanding. Just understanding basics. RBCs are red blood cells. The plasma membrane is lipid bilayer. In a plasma membrane, there consists a particular oligosaccharide sequence. What is oligosaccharide? Saccharides are actually carbohydrates. Then what is oligo? Oligo means small carbohydrates which are held together. Okay, small carbohydrates held together to form forming a little more large sugar. It is known as oligosaccharides, which is less than 20 monosaccharides. Okay, so in an RBC plasma membrane, there is a particular sequence of oligosaccharide which determines the blood group. How it determines the blood group? Okay, in the end of the oligosaccharide, if N-acetyl galactosamine is present, it will be A blood group. If at the end portion of the oligosaccharide, in place of N-acetyl galactosamine, if galactose is present, then it will be B blood group. What happens if both are present at the end? Then it will be AB blood group. If no sugar is present at the end, if no monosaccharide is present at the end of the oligosaccharide, or no sugar is present at the end of this oligosaccharide, then it is O blood group. Okay, I think you got an idea. So, and you should also remember the sequence of the oligosaccharide present in the top of the plasma membrane or present in the plasma membrane. What is the sequence? Glucose, galactose, N-acetyl glucose, then galactose fucose. This is the oligosaccharide sequence. At the end of the oligosaccharide, which sugar comes, then it determines the blood group. Look, if IAIA, both dominant form are present, it will code for A blood group, which means that if IAIA express, which means if the gene is active, they will lead to the formation of N-acetyl galactosamine, a sugar, which will bind to the RBC plasma membrane. Then it will be A blood group. If IB genes are present, it will code a protein which will produce a sugar, which is galactose, which will bind to the end of the plasma membrane. Then it will be B blood group. If no sugar are expressed, which means no genes are expressed, no sugar, then no sugar at the end of oligosaccharide, then it will be O blood group. If both sugars are expressed equally, codominantly expressed, then both the sugar will be present at the end of the oligosaccharide sequence. I think you got a basic idea. Now we will go to the structure, so you will get more and more idea. Look, this is the condition. Look about O blood group. This is the oligosaccharide sequence I have mentioned. This will be glucose, galactose, and acetyl glucose. This will be galactose. This will be fucose. And to this galactose, if N-acetyl galactosamine binds, then it will be A blood group. And if to this N-acetyl and to this galactose, if galactose bind, then it will be B blood group. So this is the condition. Okay, this is the condition in ABO blood group system and type of sugar. I think you got an idea. Now, if we look to the just for your information, I have put this second image. Look, A, B, O, AB blood group. These are the blood groups. So if A blood group is present, which means it will be having a sugar. A blood group will be having anti-B. Anti-B means antigen produced in the blood against antibodies or against any foreign substances. Antibodies will be produced in the body. So in case of A blood group, there will be no presence of anti-A, because if anti-A is present, it can bind to the same antigen of the blood and they will clump together. I have put this figure in order to understand that AB is a universal acceptor and O blood group is a universal donor, because look here, O blood group can be accepted by everyone, can be received by every blood group. Okay, and AB blood group can receive any type of blood, A, B, and O. So in order to understand this, I have just put this. Okay. Next question. Okay. Question number 5. Question number 5 is similar to that of question number 1. So there is no need to discuss anything regarding it. Next. Question number 6. Okay. In a marriage between a male with blood group A and female with blood group B, the progeny had either blood group AB or B. What could be the possible genotype of the parents? This is the question. So we can write down the situation here. A male and a female, marriage occurred, they are having children. A is having A blood group. Okay, so A is a male blood group. No. The thing is that one of the individual male is having A blood group, which means the chance of becoming A, the genotype is IA, also IA, and IA, and the small I. This is the genotypes for A. This is the chance that the male will be having. There are two chances, same as that of an individual or a female with B blood group. So what will be the condition? IB, 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 and small I. Okay. So and their progeny is having the blood group with either AB or B. So let us draw the genotype possibility of their children or progeny. They are saying AB or blood group is the possible blood group. No other blood group can come except AB or B. So let us draw the chance of genotype of AB. IA, IB. And the chance of genotype of B is IB and I. Sorry, I or IB, IB. This is the possible chance of B blood group. So only these three situations can occur, genotype can occur for the progenies. Let us go through the option and check the possible genotype. Option A, male IA, which means this is the thing, and female IB. Okay, let us cross them and check whether we are getting the progenies here, genotypes here. AB is possible, B is possible. Yes, we are getting AB and B. No other blood group are possible. So maybe option A is the correct answer. Now let's go to option B. Option B, there are two genotypes, A and B. So if we cross them, this is the possibility. If we cross them, only the chance of AB is possible, B is not possible. So that is not the answer. C also, what happens? AB is possible and IA is possible, which means AB and A is possible, B is not possible. So option C is also incorrect. I think option A is the right answer. For option D, they have taken this chance. They are crossing it. So AB can be possible, A can be possible, B can be also possible, but I can't be possible. When these gametes fuse, I is possible, which is not given in our question situation. So I can't be possible. So the option is option number A. I think it is clear for you. If you are having any sort of doubt, first go to the concept, clear it, come back. Even if you are having doubt, you should comment me or mail me. Okay? Understanding the concept is very, very, very important in case of genetics. Even if you are going for higher genetic studies also, understanding the concept, doing the questions is compulsory. Next, in an anterinum, a snapdragon, a red flower was crossed with white flower. In F1 generation, pink flower were obtained. Yes, this is an example of incomplete dominance. Yes, when we have discussed it, it is an example of incomplete dominance, intragenic gene interaction. There are two types of gene interaction, intragenic gene interaction and intergenic gene interaction. Intergenic gene interaction means the genes, two different genes get interacted and they change the genotype and phenotype. That is known as intergenic gene interaction. Genes within different genes interact, but in intragenic gene interaction, the interaction is within the allelic forms of the gene. It is an example of dominant, incomplete dominance, codominance are the example of intragenic gene interaction. So in intragenic gene interaction, which is the incomplete dominance and codominance, both the genotypic ratio and phenotypic ratio will be equal, which is 1:2:1. So from the option given below, there are 4 options. We can check which is the incorrect statement from the following. Pink color in F1 is due to the incomplete dominance. Yes, it is correct. Second is also correct. Yes, we will get a 1:2:1 ratio in the F2 generation. Next, law of segregation does not apply in this experiment. That's actually wrong. Actually, if law of segregation does not apply in this cross, we will not be getting the ratio. It is actually the incomplete dominance or codominance, which is the part of intragenic gene interaction, are following Mendel's principle of segregation and Mendel's law of independence assortment, but they show some extra phenomena, which is actually we studied it as a part of the extension. Okay, so they always follow Mendel's law of segregation, etc. But in each situation there will be a change. Like in case of codominance, both will be expressing together. In case of incomplete dominance, one can't express fully. In case of multiple allelism, as in case of blood groups, they will be expressing equally. So this is the condition. So here law of segregation should apply, but the option is wrong. This experiment does not follow the principle of dominance. Yes, it is correct. So actually we study it as a part of extension of Mendelism, which means there are certain changes observed in the nature, which is not said by Mendel. Okay, so the option is option C. Moving to the next question. A gene locus has 2 alleles, capital A, small a. This is the allele form of a gene. If the frequency of dominant allele A is 0.4, then what will be the frequency of a homozygous dominant, heterozygous and a homozygous recessive individual in the population? Okay, that is the question. So a gene is having two alternative form. We know that capital A and small a. The frequency of dominant allele is 0.4. The frequency of occurring this allele is 0.4, which means in a genome, entire genome, the frequency of occurring this capital A is 0.4. Okay, and what will the frequency of homozygous dominant? They are asking. Homozygous dominant means they are asking what is the frequency of capital A, capital A. We know that if a single allele capital A is having a frequency of 0.4, which means if both of them should occur together, then we should multiply it 0.4 into 0.4, which is 0.16. So homozygous dominant will be 0.16. The frequency to occur both the capital A together, then we should multiply it. That is the logic behind this mathematics. Okay, I will say that what is actually 0.4 means. The frequency of capital A is 0.4, which means that 4 into 10 raised to minus 1, which is 4 by 10, which means that out of 10, the frequency of occurrence of A is 4. 4 by 10. So what will be the frequency of small a then? It will be 6. The rest will be 6. 10 minus 4. Look. Then the chance of or the frequency is 6 by 10, which means 0.6. 0.6. Small a or the frequency of small a is homozygous recessive is 0.6 into 0.6, which is 0.36. I think you are getting it. I will say it once more. In the question they have given that capital A allele have a frequency of 0.4. So from there we can find out it is 4 by 10. Out of 10, 4 frequency. 0.4 frequency out of 10, 4 times it can occur. So we can find out that out of 10, if 4 times capital A occur, then 6, the rest 6 small a can occur. So from there we can find the single allele A, small a. So in order to find it homozygous recessive small a, it will be 0.6 into 0.6, which is 0.36, right? 0.36. So we got the two option. Yes. Now we should find heterozygous condition also. Heterozygous means this is the condition. Capital A, small a. So what is the condition? Capital A is 0.4 and into small a is 0.6. 6 into 4 is 24. So 0.24 is the frequency for this condition. We know that heterozygous can be exist in this condition too, right? So there will be one addition 0.24 also. So what will be the total? 0.48. 0.48 is the total. I think you are getting it. So option number B is the right answer. Okay. Capital A, small a, 0.4 into 0.6. In a genotype, there is also this combination possible. So we should add both of them. Here also we will get 0.24. So 0.24 plus 0.24, 0.48 is the right answer. We can also get to the answer with the help of binomial theorem. Here there are two possibility A, capital A, and possibility small a. So and the chance is 1 by 2. So from this equation a plus b the whole square, actually it is a square plus 2ab plus b square. You know that binomial. Okay. So let us give a as small a as 0.4 and b as 0.6. So here it becomes 0.16 a square. Here 0.36 and here A into B, which means AB, AB. 2 into 2 times we should take AB. So 0.4 into 6, which is 0.24, 24 into 2, 0.48. So actually we should take 0.16. So we got 3 numbers here. We should not add. This is not, we are not doing a mathematical question, we are doing a frequency. So this is first frequency, second frequency, and third frequency. Okay, I think you got the answer. There are 2 methods to do. If you need it, we will be taking a mathematics class for genetics. So this is the basic idea. I think you have got the answer. The answer is option number B. Next, select the correct statement. Spliceosomes takes part in translation. Second, Punnett square was developed by British scientist. Third, Franklin Stahl coined the term linkage. Transduction was discovered by S. Altman. The true answer yes. The true answer is option number B. Punnett square was developed by a British scientist, Reginald C. Punnett, in 1917. I think we have discussed it in the first part. First question, right? Spliceosomes takes part not in translation but in post-transcriptional process. Their spliceosomes are certain complex which helps in the splicing process. Splicing means the removal of introns which are a part of a DNA. After the process of transcription, they go to post-transcriptional changes in which spliceosomes helps in the removal of introns by the process of splicing. We will deal it very deeply in the molecular basis of inheritance, which I have not taken the class. Okay. Franklin Stahl coined the term linkage. No, it is not the correct answer. Transduction was not discovered by S. Altman. I'm not going much deep into what is the process of transduction. Is the process in which we know that bacteria divides by the process of asexual reproduction, but in order to maintain their genetic diversity, a mode of sexual reproduction like a transduction is maintained, in which they release their genetic material to virus, or virus takes their genetic material and spread into a next bacteria by virus, which is known as bacteriophage, virus attacking bacteria. Next, so the option here is option B, which is the correct answer. Next, the 11th question. which one from the given below is the period of Mendel's hybridization experiment? Okay, I think I have said this earlier. I think so. Mendel conducted his experiments in 1856 to 1863, like 7 years. He have before 1856 in the sense, even before 1856 he have started the experiment. It took him 2 years to select the 34 different variety of pea plant. I think you can understand what I am saying. 34 different varieties of pea plant were taken by Mendel. It took 2 years for this, and also he conducted experiment from 1856 to 1863, and he published his paper. His research result was published in 1865. He presented his result in 1865 in Brune Society of Plant Science, no, Natural Society. And in 1866 his paper was published, and till 1900 his work was not seen by the outside world. So his experiments, he conducted his experiment from 1856 to 1863. In 1816 1865 he presented his paper. 1866 the paper was published. His work was published, approved. Okay, so the option is option number A. Next, the 12th question. Which were the characters which was not taken? Yes, we know that option number B is the answer. Trichomes were not selected by Mendel. Trichomes are hair-like structures. Next, 10th. Oh, okay, sorry, there was a mistake in question number. Next, 10th question, which is in your right side. 10th. Which of the following character represent inheritance of blood group in human? Okay, yes, inheritance of blood groups in human. Yes, it is an example of dominance, codominance, and multiple allelism, right? 1, 2, and 3. So the option is option B, 10B. Next, 13th question. The genotype of a husband and a wife are given. Male and female are given. Among the blood type, how many different genotypes and phenotypes are possible? Yes, this is an example of multiple allelism. So the genotypes are given here: IA, IB, and IA, II. What are the possible genotypes? Right? A is possible. This will also result in A. Next, AB is possible. So we can check the genotype, right? So I will write down the genotype first. From the look, IA. I am doing it from the question. IA. Next, II. Then IBIA. It means AB. Okay. And IBI. IBI. Which means 4. 2 into 2. 2 into 2 combinations are possible. Okay. Okay. So 4 phenotypes are possible. We are having option number B and D as 4. Now we can check how many genotypes are possible. Look, here it will form a blood group, AB blood group, and B blood group. So 3 genotypes. Option number B. Okay. Next, 14th question. I think you are getting me. Next, 14th question. A true breeding plant. What is a true breeding plant? Okay. I think I have also said this in the part 1 of the lecture. What is actually a true breeding plant? Just listen. You know, I have said that Mendel took 34 varieties of pea plants. It took him 2 years for selecting the pea plant and to confirm that the plants are true breeding in nature. Which means, if the plants are self-fertilized, it should give the same phenotype and genotype of the parent. This is what is known as true breeding. Example, Mendel took Pisum sativum as an experimental organism. It is a garden pea or a pea plant. Usually, peas are self-fertilized. Usually in nature it occurs by the process of self-fertilization. What is self-fertilization? We know that the male sex organ is the anther in a reproductive structure, flower, and the female is the stigma, which means stigma, style, and ovary is the female part of the flower. So usually, in order to confirm a true breeding plant, what Mendel did is that he self-fertilized the plant and checked the upcoming 2 generations. If the upcoming 2 generations are having the same character as that of the first parent, then we can confirm it is a true breeding plant. which means it should show near homozygous. It should not have an extra recessive allele even in its heterozygous form. That is known as true breeding plan. Okay, so option number C is the right answer. I think you are understanding it. Okay, there is a process of emasculation. So how did Mendel cross two different plants with contrasting character? Which means he found by self-fertilization that a tall plant is always having capital A, and a short plant is always having small a. So they are homozygous and they are self-fertilized and confirmed that they are true breeding plant. So how did Mendel cross these two different plants? Is by the process of emasculation. Emasculation is the removal of the male sex organ, the anther, in the plants, so that the same plant's pollen won't get into the stigma of the same female sex organ of the flower. So that he can take an example is that he took a plant, a tall female plant, in which the flower of the tall female plant's flower anther was removed by the process of emasculation, and a short plant was taken, the short plant's pollen was taken, and it was rubbed in the stigma portion of the sex organ, female sex organ. This is how a cross is done by with two different characters. Okay. Okay. This is a true breeding plan. This is how Mendel done his experiment. Next, 15th question. Match the terms in column 1 and column. Okay. So what is actually dominance? We can look it in the graph. Column number 2. What is dominant? Yes. Look, option number 2. In a heterozygous organism, only one allele express itself. Right. This is the best definition for dominance. In a heterozygous form, only one allele can express. So A2. Next, B. Codominance. In a heterozygous organism, both alleles express themselves fully. This is an example of codominance, and this is a intragenic gene interaction. The gene interaction between same gene, same alleles of the same gene. It is known as intragenic gene interaction. Intragenic gene interaction. Codominance comes under intragenic gene interaction. Okay. Next, pleiotropy and polygenic inheritance. Yes, the two topics we have discussed earlier. Pleiotropy. Option number 4. A single gene can influence a many character. A single gene have more than one phenotypic effect. Okay. We have also said it in example of phenylketonuria. Phenylketonuria is an example of pleiotropy. If the PAH gene is mutated, the entire pathway will be blocked. This is an example of pleiotropy. Next, polygenic inheritance. We know that polygenic inheritance, many genes have a effect on a particular character. Cumulative effect or additive effect. Each gene contribute additive effect. It is known as polygenic inheritance. Environment also play a factor here. It will be having a continuous variation. A bell-shaped graph will be obtained in case of polygenic inheritance. Okay. So option number A is the right answer. Polygenic inheritance, many genes govern a single character. I have said everything very deeply in the lecture, so there is no actual need to say it more, right? We have studied it actually. So next, in the 16th, next question. A tall, true breeding garden pea plant is crossed with a dwarf true breeding garden pea plant. Yes, a contrasting character, right? Tall and dwarf. Okay, when the F1 plants were selfed, the resulting genotype were in the ratio. Yes, we know that. The question is asking about the Mendel's F1 Mendel's cross and the F1 ratio. We know that in Mendel's cross F1, he found that all the plants were tall, right? All the plants were tall. If the tall and the dwarf character were taken, the F1 were all tall. Then the question is asking what happens when the F1 is self and the resulting genotype in the F2. What is the condition? We know that in resulting genotype F2 is 1:2:1, okay, which is the genotype, and phenotype is 3:1, tall is to dark. So 1:2:1, we know that 1 indicate the tall homozygous, 2 indicate the heterozygous form, and the other one is the dwarf. So the option number D, option D. Next, 16th question. A gene showing codominance has. Please read the options. We know that codominance is an example of intragenic gene interaction where the alleles of the same gene interact each other to form an extension from the Mendelism. We know that in codominance and incomplete dominance, both the phenotypic and genotypic ratio is 1:2:1. We have found out that, right? So the option here is option D. Option A can't be present. Option A is not true in case of codominance, right? Option B, alleles tightly linked on the same chromosome. They are saying that alleles are linked. Actually, what is the definition of an allele? Allele is the alternative forms of the same gene present on the same locus on a homologous chromosome. This is the definition of allele. They can't be present on the same chromosome, but they can be present in the same locus on a homologous chromosome. Okay. So option D is the right answer. Next, in Mendel's or in his classical experiment on pea plant, Mendel did not use, yes, he took 7 characters. The character did not include the pod length. Pod length was not included, but pod color and pod shape was included. I always said you to remember a plant where a tall plant with axial violet flower. A tall plant with axial violet flower having a green inflated pod with yellow round seeds. This was the dominant character taken by Mendel. So pod length was not in the 7 contrasting character. Actually, you should also remember that there is, if we say the seed color, actually it is the seed endosperm color, not the seed coat color. Mendel also took seed coat color, and the seed coat color was white and gray. White was the recessive form, gray was the dominant form. Okay, so here the option of 18th question is B, did not use the pod length. Next, what is a pleiotropic gene? Yeah, this question has been repeated. What is a pleiotropy? A phenomenon in which a gene has more than one phenotypic effect. It is known as pleiotropy. And what is polygenic inheritance? More than one gene contribute to a same phenotypic effect, which is having additive effect or a cumulative effect. That is known as polygenic inheritance. Here the option for 19 is D, which controls the multiple traits in an organism. Okay. A good example of pleiotropy, I have said it once again, I am saying it, a good example. I think this is the 3rd time I am repeating. What is a good example of polygenic pleiotropy mechanism is in case of a disease, autosomal recessive disease. I think you know that. What is autosomal recessive disease? Autosomal recessive disease, phenylketonuria. What is phenylketonuria? Phenylalanine is an amino acid present in our body. When phenylalanine is usually phenylalanine gets converted into tyrosine and another amino acid present in our body with the help of PAH enzyme, phenylalanine hydroxylase. So what happens in a phenylketonuric organism, a autosomal recessive disease? What happens is that the PAH enzyme can't be found. The PAH enzyme is absent in case of the organism. Why it is absent? Because the gene which codes for the protein PAH, we know that enzyme is a protein. So the gene which codes for the PAH enzyme can't express. So what happens in a normal human being? Phenylalanine get converted into another amino acid, tyrosine. Now, tyrosine is a precursor or a reactant or a substrate for many other biochemical synthesis in our body. Like melanin synthesis, tyrosine is needed. It is also needed for certain hormone production like epinephrine or norepinephrine. So if tyrosine can't produce, is not able to be produced, what happens? This pathway will be blocked. Phenylalanine is converted to tyrosine with the help of this PAH enzyme. When PAH enzyme is not active or present, why? What is the reason? Because the gene which codes for the PAH gene is not expressed. This is a good example of a pleiotropic gene which codes for the PAH enzyme. So option number D is the right answer. Next, 20th question. A fruit color in a squash is an example of, I think I didn't mention anything regarding this. Look, epistasis. Okay, just remember, just try to hear. We said about intragenic gene interaction, right? What are intragenic gene interaction? We have discussed codominance, incomplete dominance. But there is also an interaction between different genes. What was intragenic gene interaction? Intra means inside, right? Within. What was in intragenic gene interaction? A gene, the alleles of the same gene. But in intergenic, different genes get interacted. Examples of intergenic gene interaction is recessive epistasis is a good example. Epistasis is divided into two: dominant epistasis, recessive epistasis. Also, the other intergenic gene interaction are complementary gene action, collaboratory gene action, duplicate gene action, etc. I did not go much into it because it was not mentioned in your NCERT, but you can hear it. First, we will talk about epistasis. Epistasis is a condition or an intergenic gene interaction in which one gene, let us take a gene 1 and gene 2. Gene 1 has an effect on other gene 2. When gene 1 is present in its dominant form, either dominant heterozygous or dominant homozygous, this dominant form can suppress the expression of the other gene. It is known as epistasis and dominant epistasis. In dominant epistasis, the ratio of phenotypic ratio is 12:3:1. What happens in a recessive epistasis? In recessive epistasis, the recessive form of a gene 1 can inhibit the gene 2. So the one who get inhibited or masked is known as hypostatic gene, and the gene which can mask the hypostatic gene is known as epistatic gene. So there are two types of epistasis: dominant epistasis and recessive epistasis. The phenotypic ratio of recessive epistasis is 9:3:4. Okay. So fruit color squash is an example of dominant epistasis. I will show you an example which is given in an international textbook. Benjamin, look, this is a squash. So actually 3 colors are possible. White color squash is available, seen in nature, green color and yellow color. So if what happens in a dominant epistasis? Dominant epistasis, a dominant form of a gene suppress the recessive form. This is the condition. I am not much going to it. If it is needed, I will be taking a class because it will be very confusing for you if I start here. For example, I will give a small example, so I think you will understand. Just if you please see this figure, there are 3 colors are possible. The squash in the color is due to the presence of an intergenic gene interaction epistasis. Okay, I will give you some idea. Look, consider a product A. Product A is converted into product B, and also it can also be converted into product C. Product A is converted into product B with the help of an enzyme E1. Consider E1. It is all product B can also be converted into another product. Reactant A can be converted into another product with the help of enzyme 2. Consider E2. Okay. For example, I am saying let capital Y. There are 2 genes, Y gene and example Y gene and O gene. I am taking just 2 examples of Y gene and O gene. So Y gene is responsible for the production of this enzyme. If Y gene is expressed, Y gene is seen in its dominant form, enzyme 1 will be expressed, and if O gene is seen in its dominant form, then enzyme 2 will be expressed. So what happens And there's other condition also. I think you are getting an idea. Small idea is required for it only. Now, just consider a situation also. Enzyme 1 has more affinity than enzyme 2 to the substrate A. Okay, substrate A. Which means if enzyme 1 and enzyme 2 are present in same condition, same concentration, enzyme 1 will have the higher binding with the substrate A. This is the condition. So, we are studying dominant epistasis. I'm now saying the cases. So, if capital Y is present in its dominant form, E1 enzyme will be produced, then A will be converted into B. This is known as dominant epistasis because E2 is also expressed there, but it can't bind with A because E1 has the higher affinity. So let us write the genotypic and phenotypic ratio in F2 generation. Okay. So capital Y. I have also said how to write the genotype for entrance method. First write the dominant form 3 together, then heterozygous dominant, then 3 times, then homozygous recessive also 3 times. Y and Y. Next, take the next gene O, write in the dominant form. Now heterozygous form, now homozygous recessive, dominant, heterozygous, recessive form, dominant, heterozygous, dominant, heterozygous, and recessive. Okay, we know that it is here 1:2, 2:1, 2:4, 4:2, 1:2, 2:1. I have said it much more in my lecture. That is the reason I am not giving it. I am not writing it. It is actually time wastage, right? So if both are present together, which means capital Y, which means this can code the two enzyme. E1 and E2 are present in the situation, but we know that only the product B will be formed because E1 has higher affinity to E2. Now what will be the condition here? In this condition, E1 is present, E2 is also present, then also B will be formed. Okay. Now only capital Y is present, then also B will be formed, right? Now capital Y is also present, it is also present, O is also present, then also B will be formed. What happens here? Capital Y is present, O is also present, then also B will be formed. Now in this condition, capital Y is present, O is not present, O is absent, which means B can be only formed. This condition, capital Y is absent, so C can be formed. Here also, what happens? C can be formed. I think you are getting an idea because here Y is absent, only the presence of O, which means C can be formed. Here no enzyme can be coded because both the genes are in recessive form though, so the A will be only in the A can't be produced or A can't be converted. So this is the condition in dominant epistasis, which means the dominant form of an allele can express, a dominant form of gene Y can suppress the other form of the gene or other gene. It is known as dominant epistasis. This is reversed in case of opposite in case of recessive epistasis, having a ratio 9:3:4. Here the ratio will be 12:3:1. Just count it. It will be 12:3:1. 12B, 3C, 1A. This is known as 12:3:1 phenotypic ratio. So this is a general idea of epistasis and the dominant epistasis. Okay, it is happening. The fruit color in squash is due to the presence of dominant epistasis, the gene interaction. Okay, next we are going to the 25th question. Please read the 21st question. Please read it. 2% with AB blood group marry, have a sufficient large number of children. A, B, B blood group in 1:2:1 mode on technique on. Yes, there is it's a big paragraph. They are asking what is happening in ABO blood group system. You know that the codominance is happening, right? So option number A. Next, 20 to 22nd. Which Mendelian idea is depicted by a cross in which F1 generation resembles both the parents? Yes, codominance, right? Yes, AB blood group. Take an example of AB blood group. So A and B can be seen together. So codominance is the example. Next, 23rd question. ABO blood group in human are controlled by gene I. Yes, we know that. It has 3 allele IA, IBI. Since there are 3 different alleles, 6 different gamma genotypes are possible, right? How many phenotype can occur? Yes. How many phenotype will be occurring? A blood group, B blood group, AB blood group, and O blood group. So 4 is the answer. 4 phenotypes can be seen. Option C, 23C. Next. Which one of the following cannot be explained on the basis of Mendel's law of dominance? From Mendel's law of dominance, yes. Please look to option C. Alleles do not show any blending and both the character recover as such in F2 generation. It is actually said in law of independent assortment. Yes, right. Law of independent assortment say that each alleles are segregated individually, right? Independent assortment. That is the thing. So option number C cannot be explained. So 24C. Next, the genotype of a plant showing the dominant genotype can be determined by test cross, right? We know that while doing a test cross, we can understand the genotype of the F1 parent or a genotype of a parent. I have also done a test cross in the lecture. If the parent is heterozygous in condition, the test cross ratio will be 1:1. What is actually test cross? Test cross is actually a part of back cross in which a parent obtained, a progeny obtained is crossed with any of the genotype of the parent. So what happens if the progeny is in heterozygous condition and we are crossing it with, if the crossing is with the recessive parent, then it is known as test cross, and if the crossing it is with the dominant parent, it is known as outcross. So in order to find the genotype of the progeny, we need to cross the progeny with the recessive parent, and this cross is known as test cross. So if the progeny is in heterozygous condition, then the test cross ratio will be 1:1. And if the progeny is in homozygous condition, homozygous can be recessive or dominant, it will be in the, will be no ratio. Every progeny, the next coming progeny will be same. This is the condition in test cross. So the option is option number A. What I'm going to do is that I will be posting this in my YouTube as part 1 and part 2 because it is harder for me to post the entire lecture of 50 questions. So it will be difficult for me. So I will be first taking a part of it, part 1 25 questions and part 2 25 in the next 25. So I will be continuing but in the next video. Okay.